Hey everyone, here we introduce FlyGM, a whole-brain connectomic graph model that enables whole-body locomotion control in the fruit fly. Let's start with the motivation. The fruit fly, Drosophila, is only 2 to 4 millimeters long, yet it walks, turns, and flies with remarkable precision. And thanks to a recent breakthrough in connectomics, specifically the FlyWire project, we now have the complete synaptic wiring map of the fly's entire brain. Around 140,000 neurons and over 50 million synapses. This raises a fascinating question. Can we take this static wiring diagram and directly turn it into a functional neural controller that actually drives a body? Here we introduce FlyGM. We build a graph neural network whose structure is taken directly from the connectome. At each time step, the fly receives sensory observations from the physics simulator, which are encoded into the states of afferent neurons. Then, the signals propagate according to the connectome structure and are finally output by efferent neurons as actions to be executed within the simulator. Crucially, the connection weights are set using the actual excitatory and inhibitory neurotransmitter labels from the biological data, so the sign and weight of every signal respects real neural physiology. Let's dig into the architecture a bit more. The connectome is modeled as a weighted directed graph, where nodes are neurons and edges carry signed synaptic weights, positive for excitatory neurotransmitters like acetylcholine and glutamate, negative for inhibitory ones like gamma-aminobutyric acid. Neurons are split into 3 groups: afferent neurons for sensory input, intrinsic neurons for internal computation, and efferent neurons for motor output. Each neuron also has a small set of trainable parameters we call an intrinsic descriptor, which captures cell-specific properties, like excitability and gain, that the connectome data alone doesn't tell us. The forward pass works in 3 steps. First, sensory encoding. The raw observation, which can be over 1,000 dimensions for the walking task, is compressed by a small encoder and injected into the afferent neuron states. Second, graph dynamics. The synaptic weight matrix aggregates signals across the connectome, and a shared neural network updates each neuron state based on the incoming signals and its own intrinsic descriptor. Third, motor decoding. The efferent neuron states are decoded into high-dimensional continuous motor commands that drive the physics simulation. Here's the key scientific question. Does the biological connectome topology actually help, or could any random wiring do the job just as well? To find out, we tested 3 baselines. First, an Erdos-Renyi random graph with the same number of edges. Second, a degree-preserving rewiring graph, which shuffles the edges while keeping each neuron's exact number of incoming and outgoing connections. And third, a standard multilayer perceptron. The results are quite striking. On the right, you can see the learning curves. FlyGM converges faster and achieves a lower error on both the action mean and the log standard deviation during imitation learning. But the real test is the control performance table on the left. At moderate conditions, 2 centimeters per second, no turning, all models perform similarly. But when we push to 3 centimeters per second with a yaw rate of 7 radians per second, the gap becomes dramatic. FlyGM maintains an angle error of just 8.3 degrees. The rewiring graph reaches 13.6, and the Erdos-Renyi random graph completely breaks down at 125 degrees. This tells us it's not just the degree distribution that matters. It's the specific evolved topology of the connectome that carries a meaningful structural prior for controlling a physical body. Now let's take a look at the actual locomotion demos. Here's the first result: gait initiation. On the left, you can see a force-directed visualization of the connectome, where each neuron's state is compressed via principal component analysis into a scalar intensity signal, colored by neuron type, green for intrinsic, blue for afferent, and red for efferent. On the right, we show the locomotion simulation from 2 camera angles, along with the live binocular visual input from the fly's left and right eye cameras. Starting from rest, the fly achieves stable and rhythmic walking within approximately 80 milliseconds. Next, straight-line walking at 3 centimeters per second. What's really interesting here is that the model spontaneously develops the classical insect tripod gait. The left front and hind legs move in sync with the right middle leg, and vice versa. This coordination pattern emerges entirely from the trained network dynamics. For the turning task, the fly executes a sharp leftward turn at 10 radians per second. You can see asymmetric stride modulation. The inner legs shorten their steps while the outer legs stretch out. And finally, flight at 20 centimeters per second. Here, the connectome policy modulates a wing beat pattern generator to control body orientation and forward speed. The key point is that the same whole-brain graph architecture generalizes from walking to a fundamentally different locomotion mode. Beyond behavioral performance, we also looked at what's happening inside the network. To do this, we designed a complex locomotion sequence. The fly starts with straight walking, then decelerates, comes to a full pause, speeds back up, and finally executes a turn. You can see the behavioral snapshots on the bottom left, covering the full 680 milliseconds. The middle panel shows the force-directed layout of the connectome at 200 milliseconds, with neurons colored by type. And the heat map on the right is the most informative. It shows how every neuron's activity evolves across this entire sequence, with each row being one neuron, organized by biological superclass: ascending, sensory, optic, intrinsic, central, VPN, VCN, descending, motor, and endocrine. What stands out is that a clear functional separation between sensory, central, and motor populations emerges, purely from the structure of the connectome, with no explicit labeling during training. This lines up well with what experimental neuroscience has found in real Drosophila brains. In conclusion, FlyGM shows that a static biological connectome can be turned directly into a working locomotion controller. It provides a genuine structural inductive bias, handles 4 different locomotion tasks, and produces internal neural dynamics that match biological observations. Thanks for watching, and feel free to scan the QR codes for the full paper and our group page.